So, hello and welcome, everyone. My name is Judith Friesl from Yordas Group in Germany, and I'm part of the LIFE NanoExplore project that is hosting that webinar today. The LIFE NanoExplore project started in September 2018, and we are now in its second year. We are 7 European partners, and the project as such aims to develop and to demonstrate the feasibility of an integrated approach to conduct biomonitoring studies, characterize exposure levels, and to elucidate possible health effects derived from exposure to engineered nanomaterials in indoor workplaces and urban areas. Today's webinar is hosted by Professor Enrico Bergamaschi. He's a professor of occupational medicine at the Department of Public Health and Pediatrics at University of Torino, and by Professor Irina Guseva-Canu. She has at the moment trouble dial in into the webinar, so hopefully she will be able to join us at the second part. If not, we will record it and you will get her presentation sent afterwards. So it's getting an exciting webinar to see if the presenter can dial in technically. But that's the choice, I think, with the digital webinars we have nowadays. Just some technical points. I hope you can all hear us okay. Enrico and I will switch off the camera then during the presentation just to make sure you have enough, the bandwidth is enough and you can hear us okay. You are all muted at the moment. If you have any question, you should have a little chat window. Please type any questions in there, and we will, depending on the time, we will answer your questions after, at the end of the webinar, or via email after the webinar when you also get the slides. Yes, so we also aim to have a second webinar as part of this project in September, but we will send out and let you know about that afterwards. So thank you all for joining, and I hand over now to Enrico.
Thank you, Judith, for this nice introduction, and welcome to everybody, also from people that maybe are on holidays due to this beautiful time. So my topic is the human biomonitoring of nanomaterials, achievements and challenges. We know that the increased availability and use of nanotechnology-enabled products raises questions regarding potential unintended hazard to humans and the environment, and whether nanomaterials need special measures to deal with potential risk. This is a key concern for the long-term sustainability of nanotechnology. However, it's also true that to date, no overt health effects in humans caused by nanomaterials have been reported. Considering that nanotechnology is an enabling technology diffused through many productive sectors, those response relationships may be underestimated by the confounding influence of other possible chemical co-exposures. With the aim of increasing the pace of hazard identification, alternative testing strategies based on high throughput in vitro and in vivo assay have been developed. Data generated by these approaches will certainly improve the safety assessment, but the complex, multifaceted, and changeable nature of events occurring at the nanobiointerfaces also means that the full replacement of in vivo assessment is not yet possible. The biological behavior of nanomaterials is not predictable only on the basis of size, shapes, and surface chemistry, whereas the material intrinsic properties determine the synthetic identity. The context-dependent properties of the nanomaterials influence its biological identity, which is shaped in part by the absorption of biomolecules or contaminants that form a corona on the surface of nanoparticles. As a result, our current testing strategy is actualized by using simplified models, different dosing regimens, and exposure routes, and these represent a compromise to understand what could happen in humans, which requires more caution in the interpretation of findings with consideration of weight of evidence. Such insufficiency and ambiguity of the existing in vitro and in vivo toxicological data on nanomaterials may hamper the risk assessment required for a scientifically sound regulatory policy and affect the reliability of decision-making tools, such as, for instance, deriving health-based occupational exposure limits. Exposure assessment deserves specific attention in nanosafety. The OECD suggested to perform occupational environmental exposure monitoring for tracking of nanomaterial movement and concentration, providing exposure parameters for alternative testing strategies, and validate computational models for prediction purposes, with the aim to minimize reliance of in vivo testing and limit our need to perform extensive, costly, and time-consuming testings. However, accurate assessment of health risks requires information on critical target organs and knowledge of toxic endpoints from studies in which relevant exposure routes and exposure levels is assessed. To overcome uncertainties due to the inherent limitation of such simplified models, biological monitoring as a tool to assess exposure and health effects from nanomaterials has recently been emphasized. Biological monitoring has been used for decades as a fundamental tool in environmental and occupational health risk assessment to identify potential hazards of new and emerging chemicals, and in health surveillance to monitor groups at risk of health outcomes. Biomarkers are recommended for assessing the effects of exposure to harmful substances, specifically where there are low or intermittent levels of exposure, mixture of toxicants that may act synergistically, or exposure resulting in disease with long-term latency. Since biomarkers are directly related to the adverse effect which one attempts to prevent, the use of biomarkers and biomonitoring represent an essential tool in health risk assessment. In spite of some issues, there are many advances in biomarker research. The ability of nanomaterials to translocate from the route of entry, and thence the possibility for the assessment of their concentration in blood or in urine or other accessible biological compartment, and their dissolution in biological media are interesting points. If a nanomaterial dissolves, it becomes a conventional chemical toxicology issue. Since nanomaterial can interact with multiple organs and affect different cell functions, it is difficult to determine with conventional assays what changes and adverse effects may occur, and whether biochemical or functional changes can be material specific or exposure route specific. Biomarkers of exposure should provide objective demonstration of the absorption of chemical in the body, thus leading to estimating an internal dose, although only a very small proportion of the inhaled dose undergoes translocation, with repeated exposure and low particle fraction showing migration with years, a considerable internal dose might be achieved. Metal nanoparticles which release metal ions or dissolve in biological media could be detectable as metal species with appropriate analytical methods, but it's also possible to find particles in biological fluids. To date, data on human are very preliminary and fragmented. By collecting and analyzing breath, we can have an estimate of the dose of particles retained in the lung. Non-volatile compounds released from the airway leaning fluids in the form of aerosolized particles can range from small inorganic ions, larger organic molecules, proteins, macromolecules, as well as cytokines. Exhaled breath or breath condensate can be collected during tidal breathing using specially designed condensers. Being collected in a totally non-invasive way, ABC is particularly suitable to be applied in field studies and for longitudinal assessment of pulmonary biology. This novel approach has represented a significant advance over the analysis of traditional media, especially when biological monitoring is not possible. For example, ABC has been applied in occupational studies aimed to the non-invasive assessment of target tissue dose of pneumotoxic metals, titanium dioxide, and respirable crystalline silica, like in the quantitative study carried out by the partners of the project EST, now Unisanté in Lausanne. Research of biomarkers have already been generated a large amount of findings supporting the validity of such intermediate endpoints to assess changes before clinically apparent disease occurs. The lack of specificity of all the biomarkers developed so far seems to hamper the applicability of biomonitoring to nano-objects. Biomarkers of interest can range from oxidative stress and inflammation, such as cytokines, isoproteins, leukotrienes, pH changes, nitrogen-reactive species, such as nitrates, nitrites, and nitrotyrosine oxides, hydrogen peroxide, aldehydes. These biomarkers have a consistent pathophysiological meaning, reflecting airway inflammation, but also systemic effects, not only acute effects, but also long-term effects. However, some biomarkers can also have a prognostic value. For instance, the fibrogenic cytokine KL6, which has been assessed in workers occupationally exposed to carbon-based nanomaterials. Since biological monitoring has a different meaning in research and practice, candidate biomarkers which are potentially useful for occupational health surveillance and epidemiology should meet validity criteria and thus undergo field validation and assessment of their predictive value towards relevant health outcomes and ultimately potential risks. To assess the quality of biomarkers, it is mandatory to consider temporal trends in exposure level, the ability to reflect internal exposure or early reversible biological changes, analytical methods kept under control by quality assurance, establishing reference range and limit values that enable the interpretation of eventual changes, and the health significance or predictive value of observed changes. Currently, there are no examples of human biomonitoring as a part of routine assessment of workers exposed to engineered nanomaterials, as well as no regulatory requirements are available for human biomonitoring. However, there is a growing body of research that is setting the stage for the use of biomonitoring to supplement environmental exposure assessment, to achieve a suitable risk assessment, and management in occupational setting. Some field studies have applied effect biomarkers in small groups of nanotechnology workers, looking at the main target organs. A systematic review identified more than 20, about 30 occupational studies concerning metal oxides, amorphous silica, carbon-based nanomaterials, and multiple exposure. These you can see some example of this. There is no time to comment, but I will leave you the slide just for your consideration. There are the two main examples, titanium dioxide and carbon-based nanomaterials. What we can conclude about this study is that all these studies have shown the involvement of at least 3 organs or systems, namely respiratory, cardiovascular, and immune systems, and confirmed the oxidative damage to nucleic acid, lipid and lipids, and inflammation as the central mechanism of action for nanomaterials. In spite of a limited evidence of adverse effects in workers exposed to the most commonly used nanomaterials, some biological changes are suggestive for subtle and mild adverse impact. However, a critical evaluation of current data on biological effects and the existing biomarkers of nanomaterial exposure reveals some drawbacks. Most of the epidemiological investigations were cross-sectional, so the validity and the reproducibility of biomarkers were not assessed due to generally small study samples used and methodological limitation of most available studies. Changes in biomarkers level compared to controls were also observed. However, limited exposure data in the relatively short period since the first exposure may have influenced the incidence of adverse effects found in such preliminary epi-studies. As a whole, the available human studies doesn't allow to get insight about reproducibility and inter-individual variability, the only source being represented by the variability in exposure scenarios. Although it is possible to identify candidate biomarkers, their suitability in terms of pathological meaning, specificity, and sensitivity towards health outcome should be assessed in prospective health studies. It has been recognized by a panel of international experts some years ago, as you know, as you see, that repeated exposure and effect biomarkers in longitudinal studies is needed to investigate whether unspecific biological changes could be indicative or predictive of clinical effects. Moreover, to provide a coherent approach and make future epidemiological research a reality, a well-defined framework is needed for the careful choice of materials, exposure characterization, identification of study populations, definition of health endpoints, and evaluation of the appropriateness of study design, data collection and analysis, and interpretation of results. Studies on engineered nanomaterials and human health are still experiencing many issues, practical issues, such as the temporality of nanotechnology development and penetration into everyday life, scientific issues requiring fundamental and applied research to characterize and classify different nanomaterials according to their toxicity, methodological issues in measuring individual exposure and investigating their specific medium and long-term effects, political and regulatory issues. At present, the last seems to be the more challenging. A pragmatic approach is to draw exploratory and candidate biomarkers from other fields of particles and metal toxicology to find similarities and differences in molecular pathways of toxicity, and to select high-value biomarkers. Ideally, these general biomarkers could be refined, and nanospecific biomarkers can be developed and tested at later stages. Strictly controls human studies considering exposure data are needed to assess the validity of candidate biomarkers, which can be further applied in observational studies aimed at assessing their predictivity towards health outcomes. Thus, working under the assumption that different nanoparticles may lead to the same pathways for disease or share common mechanisms like inflammation, the issue of nanomaterial heterogeneity could be considered less critical. By identifying early signs in susceptible individuals, occupational physicians will remove them from harmful exposures, or the owner can introduce exposure control measures for reducing or avoiding exposure. By reassessing the biological health status based on early effect biomarkers measurement, it could be possible to confirm the intervention effectiveness and generalize it elsewhere. As a whole, biomonitoring is not actually part of the classical hierarchy of risk management tools. Rather, it may help in evaluate how the controls are effective. Biomonitoring studies represent a valuable component of an integrated strategy and a proactive approach to risk assessment and risk management, and also an opportunity for companies concerned with the responsible development of nanotechnology. Thank you.
Okay, thank you. Yes, and now I hand over to Irina Guseva-Canu. She is professor at the Institute for Work and Health at the University of Lausanne.
Thank you, Judith, for the presentation. So I share my screen now for continuing the presentation. Can you see it on the full screen now? Okay, so my task is to present the practical part of this webinar after the marvelous introduction by Enrico of main principles of biomonitoring. We will see together how to implement it in the real life in companies dealing with engineered nanomaterials. As a point of departure, I would like to present or to remind some methodological consideration we should keep in mind when assessing the exposure and its effects in workers. When discussing causal inference in epidemiology, there are three key points to consider. The first is the strength of statistical association between exposure and effect, and ideally the demonstration of a dose-response relationship. Both are a function of statistical power and the sample size, which should be sufficient enough to get enough of statistical power. Because we know that in single companies, the work size dealing with engineered nanomaterials are usually small, we should go for the multicentric studies and working with multiple companies to do something meaningful. The second key point is the temporality between the exposure and the effect. So ideally, to speak about causal inference, the exposure should precede the effect. That's why we should go for the prospective follow-up with at least 6 months to see any effect of the exposure. And the third key point is the specificity of the effect to the exposure in relation to the exposure. And we know that the effects that are currently studied in nano workers are not specific of nanoparticles of engineered nanomaterials. So it's possible that there are plenty of confounding and effect modifying factors that should be assessed in addition to the measures of exposure and effects by other methods like questionnaires or additional measurements. So for dealing with these key points and be able to discuss causal inference of the effect we expect to observe in our study, in our biomonitoring study in nano workers, we should prepare thoroughly our study protocol, and in the frame of NanoExplore project, we conducted 3 preparatory actions. One was to assess and identify the most valid and the most convenient biomarkers of exposure and health effects. Also to identify the set of exposure measurement devices that we could easily bring and operate in the companies dealing with nanomaterials. And third, to assess the diversity in working conditions and in company organizations where we hope to go to proceed with these measurements and enroll workers and do the prospective follow-up of the health effects and exposure. And for this, we conducted an online survey. We called it NanoExplore Survey. It started last year in spring, and actually we sent an invitation to more than 1,000 companies dealing with engineered nanomaterials, which were previously identified by Yordas. So, and we got quite a small response rate, around 2.5, which is actually normal according to the literature. So from the companies which answered to us, we addressed some common features which could characterize the working conditions that we could expect today in the companies producing or handling engineered nanomaterials. The first of all, the first interesting information that most of companies actually produce less than 5 kilograms of engineered nanomaterials a year, which is not a big amount of production. These engineered nanomaterials or nanoparticles are handled in different physical forms, which could be solid, liquid, or aerosol. And in most companies who answered to our survey, there are already a lot of solutions to control the exposure. So the information we got from these 3 questions is actually that we could not expect a high exposure potential and high exposure levels, but in contrast, we should be prepared for the moderate to low exposure potential with quite low exposure levels. So this means that we should have a sufficiently large sample size and plan quite a long follow-up to see any effect of this low level exposure. The second interesting information was about the processes related to engineered nanomaterials. So the survey showed us that an average during a day, companies dealing with engineered nanomaterials, either in producing them or in transforming them or in incorporating them into composite matrices. And this process actually lasts less than 4 hours in most companies. And they do it less than 4 days a week. And these processes actually involve less than 10 employees in half of the companies. So from these questions, we remind and retain the following information: that we could actually expect to conduct 1-day surveillance during an 8-hour shift, twice during a working week, starting with the first day and ending with the last day of the process. And we should be prepared to manage individual data collection and biological sampling simultaneously for up to 5 workers. So this requests a good planning of the study, good logistical preparation, and of course, underlines the importance of the study coordination. Another interesting part of this survey covered the propensity and the reasons to participate or to refuse the study participation by companies and workers. And actually, as you can see, most of companies, 72% of our respondents said that they are willing to participate in a biomonitoring study. we asked for which reasons companies which do not want to participate refuses the participation. And the first answer was the concerns about data protection and confidentialities. But there were also other reasons, which, for example, could be the participation in other studies. We had, for example, some American companies who said that they were already involved in the similar studies, or that they have enough of knowledge and enough of business with safety and health at work. But what was interesting that the reasons to participate were much more various, and it went from improving the safety of workers, increasing the knowledge of engineered nanomaterial related safety and health practices, till the providing data useful for the European REACH registration. So, and when we asked what would motivate workers to participate in the study with biomonitoring of exposure and health effects related to engineered nanomaterials, we actually asked first the managers and after the workers themselves. And the managers said, most of them, well, what's important to underline here is that 50% of management could not answer the question about the workers' motivation to participate. But those who answered, who were another half, actually cited different reasons. And that the most cited one was that participants would appreciate to receive individual results regarding their exposure and also biological data. They would also appreciate to have health examinations and receive the results about the project, but also that probably it would be beneficial to other workers in the sector, not only in their company. And what's fantastic that workers actually cited these three first reasons as the motivating factors for their participation. And they also cited another one, which was not actually mentioned by the managers, that they would more participate if the project would be funded by a public funding structure. So from this part of the surveys, we can retain the importance to ensure the confidentiality of participants and of the data protection, which is one of the reasons which hampers the participation. That it is important to ensure the company result communication and discuss these results with companies, and also to deliver in an appropriate way individual results, because it's really what motivates workers to participate. The third part of the survey covered the conditions which would be the more appropriate for conducting a biomonitoring study in a company. And managers and health and safety engineers actually were mostly comfortable with conducting exposure monitoring, but less comfortable with conducting biological sampling among their workers. Actually, the reason for that, when we ask, do you have a room or some amenities for conducting biological sampling or medical exams or questionnaires, etc., they were very few to answer positively to this question. So, and regarding the campaign duration, mostly said that it should be conducted between April and June or September and November, which are the best campaign months, and that we should really avoid winter, Christmas period and also summer period, which are periods of reporting or some production series. So they mostly said that the company should last less than 7 days and request less than 1 hour a day for sampling procedures, for example, and it should really take place during working hours. We said also the same question to workers, and all of them were comfortable with both exposure monitoring and with self-administered questionnaires, and all were comfortable with exalt air sampling procedures. But with respect to urine, exalt breath condensate, and buccal cell collection, they were quite comfortable, but less than with really non-invasive exalt air sampling. And as manager, they preferred that biological sampling procedure lasts less than 1 hour a day and that it takes place during working hours. So from this information, we decided to propose a study which would have a multicentric prospective cohort design with an intervention component. I will explain it later. So in this scheme, you can see that we plan to first sign an agreement or a memorandum of understanding with the company and discuss when exactly it would be the most appropriate for the company to receive us for an information visit, which is planned to provide the information about the study, the information about the study benefits, and to answer to the questions coming from workers, because their participation is important and we need to be clear with them on what we are going to do and what result they will receive. So after we have this session, we expect to receive signed content forms from workers and start our campaigns. And the study is has a prospective design, so we plan to do 2 campaigns. One is so-called recruitment campaign, and another the follow-up campaign, which would have place after 6 or 9 months of follow-up. So 2 campaigns have quite a similar organization. Each campaign would last 4 days during working time, of course, as the NanoExplore survey suggested us, and we will do the pre-shift biological sampling and post-shift, and the last day, 4 days later, biological sampling from workers. And the exposure assessment would last during the full 4 days of the campaign. So depending on the results, we would provide some suggestions in how companies could control the exposure and reduce the exposure. And during the follow-up period, we could see the effect of the exposure reduction, which could be measured during the follow-up campaign with pre- and post-shift sampling of biological matrices and the exposure assessment during 4 working days again. We would have 3 groups of participants. The first group, called exposed, would comprise workers exposed or dealing with engineered nanomaterial, who manipulate them, who handle them. And we can also consider the internal control group, which would comprise non-exposed workers, workers working in administrative departments from companies where the workers from the exposed group are recruited. And finally, we have a third control group, which is called universal control group. And this group will comprise workers from companies with confirmed absence of exposure to engineered nanomaterials or incidental nanoparticles. Why we consider these 2 control groups? Because from the literature, we see that clearly even non-exposed workers in companies dealing with engineered nanomaterials, these administrative workers are often also exposed from air surface contamination, from contamination through clothes, for example, so they are not really not exposed. So with our third group of universal controls, we should be sure that we have really non-exposed workers. So I said before that we should have a sufficient sample size to be able to demonstrate the exposure effect association and the dose-response relationship, ideally necessary for causal inference conclusion. So we calculated sample size based on data from the literature. We consider, for example, data from HOPE in a similar project measuring similar biomarkers as we plan to measure here in NanoExplore. And we derived from them some standard deviation within groups for urine and for EBC to see how the effects varies, this intra-subject variation within groups. And we consider Pelklova studies for see which are the differences that are currently observed between exposed and non-exposed groups in terms of effect biomarkers of effects. So in urine we have this shape of the curve, and in exhaled breath condensate it's like that. And for the difference, local difference between biomarkers measured in exposed and unexposed workers is plotted on the x-axis. So we can see that to observe a difference of 0.17, which corresponds to the 50% difference, we need a sample of 40 workers. For a difference in the scenario B, which corresponds to 30% difference between exposed and unexposed workers in a biomarker, we actually need 75 workers. And for the scenario C, which corresponds to the 20% difference, we need only 120 workers. This is for the difference in urine biomarkers. And we made the same calculation with a standard deviation which is lower, and we need less individuals in our sample to see the 25% difference in EBC variation of biomarkers. So it requires actually less than 100 workers. And because we are also interested in the effect of exposure reduction during the follow-up, we did similar calculation to see how many subjects we need to observe the difference between external exposure and the effects in internal exposure biomarkers between exposed and non-exposed groups. And actually we end up with something which is less than 200 people for the scenario C, which corresponds to 60% difference. To conclude with this different calculation, we decided to include in our sample size at minimum 160 people with 80 exposed workers and 80 controls. And this number actually allows us to expect the effect difference of 25% and also account to 25% of loss to follow-up, because according to the literature, people renounce to participate in the study during the follow-up, and sometimes we lose participants for different reasons. So this is about a sample size, and now what we will measure in terms of exposure. In NanoExplore, we have how can I say, our NanoExplore device, which actually comprises 3 different devices, and they measure the exposure extent for different fractions of particles in terms of mass concentration with OPC device, which is miniaturized and comprised in it, the number size and the lung disposal surface area for particles, which is measured with partector, which is here in this box. And we also have some pumps to pump the air on the filters and to collect particles on the temp grid to do chemical and physical characterization of the particles in the ambient air. So this device is used for the stationary exposure assessment, but they could be each of them extracted for individual exposure assessment in the participant breath zone. And along with this exposure measurement device, we would collect company characterization by questionnaire during our information visit, and some contextual data like temperature, hygrometry, and also notify different events and tasks which would have place during our 4-day campaign. So now what individual data we will collect? I said that because we are dealing with non-specific effects, we should collect a lot of information for assessing for confounding and effect modification factors. And most of them will be collected using the self-administered epidemiological questionnaire. So we will ask about the exposure determinants, smoking status, health status, allergies, some life habits, etc. And we will also do medical exams and a pulmonary function test to measure the volumes and flows rates, and measure the environmental oxidative potential to be able to standardize our oxidative biomarkers measures and also oxidative potential measures in the exhaled air. So, and regarding the biomarker of exposure and effects, they are tabulated here. So we will collect 3 biological matrices, the exhaled breast condensate, urine, and exhaled air. And in urine and in exhaled breast condensate, we will measure the biomarkers of exposure, which are mostly metals, because our nanomaterials are mostly of metallic or carbonaceous nature, and the biomarkers of effect focused on cytokines and pro-inflammatory interleukins, for example, immunosuppressive interleukins, allergy-related biomarkers. Leukotrienes, clara cell proteins in EBC, and also the 3 biomarkers of oxidative stress, together with hypersensitive C-reactive protein, which is a marker of vascular inflammation. And in urine, in addition, we would measure the total antioxidant capacity and creatinine to adjust for the other measurements. And finally, we have our in-house biomarker, which is under validation as a diagnostic biomarker for respiratory diseases. It's called OPA, oxidative potential in exhaled air. So it measures all oxidative stress in the airways. And it's actually a very simple measure, but with a complex device behind and complex technology, but which lasts only 6 minutes and provides directly the result. So, and we will enter all these measured results, and in particular this direct measurement OPA using our RedCap software. So we have a license at the University Hospital of Lausanne, and we will use it for our campaign. Because as you remember, the survey showed us that data protection and data and confidentiality of participation are very important concerns for companies and also workers. So we need really guarantee an adequate protection of data and confidentiality of participants. So here it's a quite a complex scheme, but it will make things very easy at the end, because most of data from individual participants will be coded before entering the system, and the data on the worker registry will be separated of all data contained in the RedCap system. So, and what comes in paper format, for example, company questionnaire or some contextual documentation, could be coded later and entered in the server at Unisanté in the final database. But most of information will be coded during the campaigns and entered directly. And what we will measure in terms of biomarkers, once we have our samples of EBC and urine, they will be sent to UNITO in Turin, and under the supervision of Professor Bergamaschi, the analysis will be done, and the results will be sent and uploaded using the RedCap, connected with good participant numbers, and at the end we would have a complete database with no risk of loss of information, all kept confidential, and all numerized and ready for analysis. So how you saw that it's important for companies and also for workers to receive the results, and we think that it's really important to communicate the results in an appropriate way. So to communicate the company results on exposure, for example, we plan to do the reports of companies, individual results, but also the overall results during the NanoExplore study. And these results will be checked and commented by a certified occupational hygienist, experienced, and a specialist of engineered nanomaterial processes, related processes. If necessary, if, for example, in some company we will observe some very high exposure levels, this certified occupational hygienist could provide specific advices and adapt on adapted exposure reduction strategies. And it would be possible to implement these advices on exposure reduction strategies during the follow-up period and to assess the effect and effectiveness of these strategies in the last campaign, follow-up campaign. And for the workers, we will provide the results for the pulmonary function test, creatinine, and also biomarkers, together with the reference or guidelines values. And you could answer, yes, but for most of biomarkers, there are no reference values because these are still experimental molecules which are not introduced into clinics. And we incorporated a sub-project in NanoExplore, which means to assess the reference ranges for our biomarkers using meta-analysis of the existing publication. And actually, we analyzed the data for 4 biomarkers, and for other biomarkers, the results are ongoing and should be ready before we provide the results. And for the OPA, which is our new biomarker, we are conducting currently an experimental study in a representative sample of Swiss general population. So each individual report will be checked and signed by an occupational physician. And the reports will be sent directly to the participant personal addresses with a label confidential. So it will be a clear separation between professional and private result restitution. And we offer the option of a Zoom consultation with an occupational physician for those participants who will ask for it or who would need some explanation of their results or some medical advices. And for this protocol, with all these features inspired by our NanoExplore survey, we received the approval from national ethics committees in Italy already. It's ongoing in Switzerland. We have a temporary approval with some modifications on the format of a consent form. And we are awaiting for these approvals in Italy and Germany as well. So this is what I wanted to present you, this protocol under approval, and I hope that you will receive it favorably and accept participating in our study. Thank you.
Thank you very much, Irina. This was very interesting. Yeah, and with that, we close the webinar for today. I don't think that there are any immediate questions. But of course, you can reach out to us if you're interested in participating, if you want to follow the study because you find it very interesting. You can email us. I think you all got the info@LIFE NanoExplore email address, or you email, of course, Irina or Enrico directly as well. And I mentioned at the beginning, as Irina already showed a little bit the device, so we will have another webinar in September where we will go into more detail on the exposure measurement and on introducing the device in more detail. So thank you very much, everyone, and we will send you the recording towards the end of this week. And I hope to see you soon at the next webinar as well, and with a lot of interest in the study as well. Thank you.
Thank you. Bye.
Bye.